Good morning, everyone. I was softly chiding Dr. Morris about not having a recent bio, but that's okay because he'll tell you all about the work he's doing. And I know for our regular attendees, he's certainly not a stranger to any of us. But he's going to be talking about something that we haven't heard much about, and I think there's a lot of interest. But let me introduce Dr. Morris. Sheldon is a board certified family medicine doctor. He has been at UC San Diego since 2006 and is involved in clinical practice, teaching, and research. His research has focused in the past on infectious disease epidemiology and has been interested in the epidemiology, natural history, and prevention of STIs and HIV. Um, and I'm just going to go off on my own whim. I've worked with Dr. Morris on several CHRP funded studies looking at HIV prevention strategies, including PrEP. Um, and he is also one of the directors or co-directors in the UCSD. Oh gosh, I'm going to get the master's program where he teaches a course called patient-oriented research, which I also had the privilege of taking many years ago. But I'm going to let Sheldon tell you about a lot of his new work and sort of how it applies to what he's going to be talking about today. Sheldon,
Yeah,
I'll keep an eye on the chat for you, okay?
thank you. Yeah, so hi everybody. Yeah, and yeah, I've been here like Jill said, almost 18 years. So you're probably wondering who's this guy that I haven't seen for a little while? But I'll tell you what I've been up to. I'm still at the AVRC, but I still have projects. We're doing another project, and we'll talk about one day too about trying to build capacity for injectable PrEP in Southern California. Because that's sort of one of the things, and we're still looking at some STI things. But one of my bigger focuses now is being director of Sanford Stem Cell Clinical Center, which I'll talk about a little bit later, which is really developing regenerative medicine approaches to different diseases. And of course, one close to my heart is HIV. And so we have the opportunity here to have a new study that we've already started, which is a CAR T approach to HIV cure I've got City of Hope up here because that's what our collaborator is. And I'm going to show you all the people that are here, but there's Davey Smith, there, Michael Choi, Kireen Dubé is also on this project. So let's start though, a little bit what we're going to do. We're going to talk about, stem cells for HIV cure. And I think we've all heard about that. I'll just quickly cover that, how that leads us into other stem cell approaches, and in particular how we can use CAR T, which are our cellular therapies, to address HIV cure, and then we're going to go talk about what the particulars about the study that we're doing here. So we all know about the cured patients, Berlin patients, and on. This is always big news whenever it comes about, and it's always very exciting to see. And I guess the point being, if we can cure them, we can cure other people. And that's kind of the way I take it from it. I mean, maybe not everybody can have a stem cell transplant. I'll tell you why, but it gives you hope that we can do something about a cure. And stem cell placement here did in fact work, and that goes back to 2009. So that's not a good record for people in what is that, 15 years? But what the premise is, and you probably have all seen this because I think it's been covered elsewhere, is basically people are getting treated for a malignancy, they usually like lymphoma or leukemia, and they are receiving a matched allogeneic. That means somebody else is donating their bone marrow or stem cells, and then they're eradicating the disease with high dose chemotherapy and any sort of cancer treatments, replacing their bone marrow with this other person's stem cells, and those stem cells happen to be deleted for CCR5, which we know is one of the primary receptors for HIV. At least, and we see all new infections pretty much have been R5. Obviously, some do transmit, and then we did work years ago on dual tropism. But this seemed to have really worked for these patients, and those people happen to not have a CCR5 receptor with no untoward, issues related to that and their health. And then these people survived their transplant, and the reconstituted, blood that they got and immune system could not get infected by HIV. And so functionally, they have been cured. Here's just the data from the original patient, and you see here that after treatment interruption, they move on to having no detectable HIV, and so this is pretty remarkable at the time that we could see that we could do that. And you might ask, well, why don't we do that for other people? Well, again, and I think the key point is that they're being treated for leukemia and lymphomas, so they have sort of a risk of not making it. But what if you just were everybody got a stem cell transplant? So I found this nugget in terms of, if you did a transplant for non-malignant disease, what would the success rate? There still is a pretty high mortality rate related to bone marrow transplant. So we're talking, that a 10 to 30% chance with a matched sibling, and with an unrelated donor, that drops significantly. So that is just an unacceptable risk for an HIV cure strategy, because you're just not, we cannot accept pretty much any mortality for that, or very low mortality for any approach. For leukemia patients, those numbers are even lower. So even though we see these successes, there's also people that didn't make it through and didn't even survive. So that just is not a long-term strategy that can be done. And one of the issues here, there's been other studies where they actually just did a stem cell transplant with non-CCR5, uh, you know, um, people that were wild type, then were expressing CCR5, and they followed those people along, and all of them, this is three people, all of them did in fact have HIV breakthrough after their interruption of antiretroviral therapy. And this one, in this particular just piece that they talked about in Science, they were suggesting some other things that you might do in terms of stimulating the reservoir and using cytotoxic T cells, or using maybe other sort of genetic tools to get rid of HIV, or silencing that there, you can, when you do gene therapy, there's different ways you can do. You can create a gene that silences the gene you don't want. You can try to replace the gene that maybe is defective, or you can try to delete the gene through these different nucleases to cut out that gene. So those would be different approaches. But the key point there is that even if in these patients that receive these transplants, they significantly reduced the actual number of immune cells, and presumably the latent reservoir, by about 100,000 fold. But still, when they have treatment interruption, they still relapsed and had viral breakthroughs. So again, that is not going to work if you just did plain stem cell therapy. So this brings us to sort of, well, what other sort of ways can we use stem cells and gene therapies to try to get at this cure that we see in these other stem cell transplant people? And there's a few different ways. I mean, we obviously, this on your left at the bottom, you see allogeneic transplant with CCR5 deleted, double, so they have two genes, obviously, and both of them are not there. And that's our successes, right? There are four people. There's other ways to do transplant. Those were less successful when they're not deleted. I don't think autologous really works unless you manipulate their cells and give it back. So then there's this idea that maybe you could do an allogeneic transplant where you delete the CCR5 from the autologous cells, put them back in. So that is something that's sort of an active line of investigation. And then other things where you can, modify some of the cells, specifically the immune cells that target HIV, which are CAR cells. I'm going to explain what that is, where you transduce them, give them reactivity against HIV and HIV infected cells, and then they would, theoretically go and destroy those cells and clear up the HIV. Because a lot of, like, say, for example, when you have latent controllers, that some of that is driven by CD8 cells and immune mediated cells. Now, I'm not an immunologist for sure, and if immunologists want to speak to it more, they can. But that's kind of how I understand it, is that there's a strong T cell response for some of those folks. So that also leads to some of the underlying, evidence to support this approach. Now, there's also things to try to do. You could autologously transform them. That's in the upper right-hand side. You could, change the stem cells of the person and give them back, and that's the CD34. So 34 positive cells are usually the stem cell, and that's often what we use as a marker for stem cell population. So I'm going to talk about some of these the current studies that have been done with some of these newer approaches and see where we're at. So what's a CAR T cell, right? So CAR stands for chimeric antigen receptor T cell therapy. So you're genetically modifying the T cells from the patient and giving them back to them. And then that T cell has been manufactured to react against a particular antigen. So there's 6 products already approved that are FDA approved that are CAR T therapies, and that are all for leukemia and lymphoma. So in these cases, we can have an antigen that's either unique from the cancer or to that cell line or that cell group, and create a CAR T to it, and it'll wipe out those cells. So that may leave them a deficit of all the cells that have that particular antigen, but it also cures their disease. So we have a number of these, and so it's not a completely wild new area. That's something that has been used, but treating cancer is much different than treating HIV for cure. In cancer, we're dealing with something where these people are facing mortality related to their disease. HIV, we know we can control very well. So there's certainly there's a bit of difference there, and we want to show if this is going to work, it's got to be safe. How are CAR Ts made? Well, CAR Ts come from you. They come from the person, the patient. They get a leukapheresis. Those are then gone through processing where they can select out the cells that they want. In this case, we might be selecting out T cells and looking at the T cell receptor, and they may be particularly, then you can sort of isolate that population of cells, and then they go through some cellular processing where they are, given here in case there's zinc finger nuclease or CRISPR technology, and those are the things that can, edit the actual DNA in there. And that's how we can introduce. The other possible way to do it, of course, is you do a lentiviral vector. We use viruses to the cell to introduce new genes. So those are the different ways that we can manipulate those T cells. And then we grow them up, and they're processed so that they become ready for transfusion. And this is where the quality control comes in. And we actually have cellular therapy labs that work on these things. And then they can be given back to the patient, and then we look for response. So that's how the CAR T works. Again, it's not new, actually, entirely, that studies actually have been done in this, and this is sometimes called first generation type ones that they are using CD4s that have been modified with this CD3 zeta chain and then a receptors that might recognize HIV and they've been given to demonstrate safety in these 4 studies, and they were found to be tolerable. The issue was that they didn't really have any response. So even if there was response, it was very short-lived. So it showed that you could produce a CAR T cell, give it back to someone that you infected, and generally they were well tolerated in these studies. Because the big concern is adverse events related to this, and I'm going to get to this, and they're not minor. For people that receive CAR T, at least in the leukemia lymphoma setting, there's this massive, kill of those cancer cells. There's a lot of cytokines released, and there's some high toxicities that are seen in those. Here, we didn't see as much in this setting, which is promising and leading us to do other studies that we're not going to see those big cytokine release issues and the neurotoxicity that have to be managed particularly in those cancer patients. And of course, we're getting better and better at that because these products are being used in clinical practice at centers such as City of Hope and UC San Diego, and so we have expertise in that. So what's next? And sort of the next generation of these CAR T therapies is to be a little smarter about it. And here are three different kind of approaches that you might try. So again, you can manufacture the CAR T, and we've heard a lot about, and our center and Dr. Richmond, other people have done a lot of great work with broadly neutralizing antibody identification, et cetera. And those, so if you could create a broadly neutralizing antibody as the, what we can identify the HIV, then that would target most HIV. That's the idea of the broadly neutralizing antibody. So that using the BNAb as the sort of antigen, the outside part, it would be a good point, or the target of that BNAb. The also would be, should you edit the CCR5 in the T cell itself? Because if it's a T cell, it can also be a CD4 expressing gene T cell, and that will be infected by HIV. So should you protect it? So sometimes you're thinking, no, maybe that should be edited as well. Although, you want to be as simple as possible. Being too eloquent would be maybe a problem if you cut too many things out, try and do too many things like that. I always try and be the simplest approach that you can do. The issue, and I'm going to talk about a key study that's already been finished with the BNAb approach, is that you can get viral escape. And so the other idea would be, well, okay, maybe one, HIV targeted response is enough. Have two. So then you have a T cell that, maybe has can react against two different GP120 sites. And then there's also convertible T cells. People are working on this, where you can actually, or a switchable T cell where you can manufacture new T cells and actually have a stock of T cells that you could then produce. So that's another thing that's being. I don't see any studies yet that have done that. And then the other thing would be you add in some sort of reservoir, latent reservoir activating agent to try to make the HIV visible. Because as you know, HIV is a latent virus. It can hide in the cell. The cell may not even express any receptors that are targetable. And so that would be a problem, is that, can we get rid of all those latent cells? Obviously, from those cure patients that did receive hematopoietic stem cell and full ablation of their current immune systems, it did work. So I guess we can, but can we do it in this way? I don't know. So I'm going to talk about how the BNAb approach here. This is a study that's been completed and published. It was done in China, started in 2017. And again, just goes through that process of how they did it. They did the CAR T preparation, infusion, and then they did a treatment interruption, or that's what ATI stands for, ART treatment interruption. And then they looked for viral rebound. And if they rebounded, they would restart their antiretroviral therapy. Now, this one in particular used VRC01, which was from what I've read, it hits about 90% of HIV strains or variants. And so they thought this was a good target. I think this was also used in a vaccine study, if I remember correctly. They used the CD8 isolated from PBMCs. They were transduced so that they would have this target with a VRC01, and which has a GP120 specific broadly neutralizing antibody. The other thing that also came out of some cancer research, that why the first generation failed, was that there's a co-stimulatory domain that's added, this 1BB co-stimulatory domain, that activates the T cell more. So this is another advancement that came in and pretty much is incorporated in most of the new CAR T therapies. They also had some suicide genes added in, like thymidine kinase, so that you could destroy the cells if you wanted to, especially in this early phase. You wanted to have a rescue plan. So they did 15 patients that enrolled. One patient, they weren't able to manufacture the actual CAR T cell, so they weren't able to receive treatment. So they ended up with 14 treated patients. And then afterwards, of course, they had to meet certain criteria around responses of CD4 and viral loads. And 4 patients didn't meet the criterion, and 4 patients could get interruption, but they didn't want it, and they decided not to do it. So that was up to them. And 6 patients decided to have a treatment interruption and went forward with that. Generally, in this study, again, it was well tolerated, and there was no serious adverse events reported in this publication. And they did see, in this case, that viral NA and intact proviruses were decreased. But unfortunately, they did rebound, and what happened was that virus was able to work around the VRCA1 BNAb target and was able to then again, as it is very slippery virus, is able to uh, you know uh, replicate again and get around that restriction. This is just an example of the patients that they had that did do the treatment interruption and when they actually rebounded. And so pretty fast. So we're talking, here 2 weeks to pretty much 8 weeks that things worked. So on the left, you see viremia. On the right, it's, time to event of when they failed viral suppression. Um, and pretty much everybody by 10 weeks has failed here. So um, here's some more data from that study, just showing one patient. This was patient 15, and just kind of what it looked like. The um, See, the red line is the showing that the presence of the actual CAR T cell, the RNA of it, and the expression of the GP120 on the left-hand side in blue, and then on the right, the plasma HIV levels over time. And so there was, again, quickly rebound, even after the gray area is showing the time of where they were off antiretroviral therapy. So then they would have started again there at week about five weeks, I guess. The other, the proviral DNA is sort of like part of the reservoir HIV. So that was why it's important to show if that was being affected. And here we've also saw that those things were reduced in all of the patients that did do the treatment interruption. But as we had pointed out earlier, unless you're very much significantly reducing the latent reservoir, probably to zero, you're not going to achieve full cure. This was actually the adverse events reported, which I'm actually still surprised by how low this is. They even said it was non-treatment related events, which I'm again not sure. But it was grade 1 fevers, so gets us to some of the issues. So in the cancer studies, fever is a sign of cytokine release. And so the fever usually means that person gets evaluated more and whether they're getting cytokine release syndrome or do they have an infection. Obviously, there's different causes of fever. So a grade 1 fever is very low grade, and they in currently practices would be they'd be admitted and worked up, and that would be, if that was the end of it, that would be the grade 1. Higher levels of cytokine release need infusions of tocilizumab to block. That's an IL-6 blocker to reduce it. Everybody's familiar with that because it's used for COVID, but actually that is the main drug that was used for cytokine release syndrome. In the cancer studies, the rates of at least a grade 1 CRS is as high as 50%. So it's not, that's pretty significant. The other thing that it does is create neurotoxicity, so people can get confused, can get seizures. Um, and so that can be as high as 20-30% in these studies. But they didn't see this at all. But those would be the main concerns. So in fact, everybody goes on prophylactic Keppra as anti-seizure agent, and let's watch closely for CRS in our study, and that's standard therapy for any CAR T therapy. But the data from this study and those previous ones relieve me a little bit of that worry that we could see some serious adverse events, although it's always possible. It doesn't look like right now that we would expect to see that kind of level, because if they did, that'd probably be unacceptable, right? To have a lot of high grade CRS or neurological complications of something for an HIV cure. So this next study, I guess I was going to go on to some other of the current studies now that are going on. There's 3 that I know of, and I'll talk about those in particular, one of them being ours. So this one follows that if you treat this thing finger CCR5 deleted cells, and you gave those cells to uh, like sort of an autologous transplant, you see a decline, a delay in viral rebound. So from that, this is the University of Pennsylvania folks and their slide, and this is their study where they've devised this CAR T therapy. Here's it on the left lower hand side, what that actually looks like. We talked about that co-stimulatory domain that's been added, and that zeta chain addition, and then they've got just expressing the CD4 on the outside of the cell, and then they've also done the um, I think is this where they also have done the CCR5 deletion. And so the idea in the lower hand, obviously, is that they can't be infected, therefore they would remain and be responsive against HIV infected cells. They have two main cohorts, and then they're looking at treatment interruption either day one or 8 weeks later. So this is ongoing. We have no results for that. And of course, there's like criterion for when they would do the treatment interruption. They have to have a viral load less than 1,000 at the end of step 3 there when they have their treatment. And then they're just followed up. The other key point about the current expected follow-up for patients who have cellular and gene modified therapies is and stem cell therapies up to 15 years. So you're sort of committing to a long-term follow-up. In the later stages, it's not complicated, maybe once a year, but still that's sort of an FDA requirement for that. So this is one approach that is being tried. I think what's unclear is whether or not there's enough anti-HIV effect of this, but that's what they're doing. There's another study at UCSF, and that was done, I think, developed some technology from Davis. And this is that dual CAR T approach. So they're doing CD4, CD8 T cells transduced with the lentiviral, which have two different targets of the GP110E. And then the idea here is, of course, is that the one of them will interact with the GP120, then opens up another epitope that the other antigen will work against. So here, it's trying to beat so that they can. I guess this is why we, of course, did triple therapy, is that if you do maybe more than one, then you have a better result, and they're less likely that the virus can mutate around that restriction. So that's kind of the approach here being done at the UCSF study. They have 3 different cohorts, and it differs in terms of dosing of the number of cells and also whether they are conditioned with cyclophosphamide. So conditioning cells, why do we do that? So this is basically like a chemotherapeutic agent. So why would you do that? Well, you're kind of making room in the marrow for new cells to take hold, or you're creating areas in the niche that cells can move in and set up base. Because we also want to see these cells to continue to stay there to suppress any, it can continue to work against any sort of latent virus reservoir. So if you have persistence of the treatment of the cells that are being infused, then that is the long-term goal. So providing preconditioning, and we've seen this in other, because I've helped with some stem cell studies, and we've definitely seen a movement sometimes that they needed to do this conditioning point to allow for actual for the cells to move, and also reduce the risk of sort of reaction.
Oh no.
Did he just disappear?
Yeah.
He's like, poof. Oh, okay.
Well, let's hold
That
on.
was unusual. Hopefully he's rejoining. I hope he didn't have like a power outage. Anyone else experiencing power outage? Um, sorry, everyone. How come you came on, Marvin?
Well, I was just gonna say I also noticed just to make sure. What do
Wait,
you
what? Yeah, how did it come onto you all of a sudden?
Oh, could I unmuted?
Got it. Okay. All right, he's coming back on. Hope everything's that was so weird. Like it wasn't like it froze or anything, you know? Well, while we're sitting here, Michael, thanks for your question. Oh, his computer shut off. Um, okay. So, Michael asked a question that I was going to save for the end. Obviously, our speaker's not on. I don't know if anyone else on the call who is involved in this work has any thoughts on this that we can just chat about because we may lose our ability to chat at the end, depending how long this takes. I could read the question out loud.
Sorry, I'm back.
Oh, he's back. I was just trying to fill space. Sorry.
Yeah, I guess I had it plugged in, but I guess it wasn't charging, so
Ah,
it shut down on me. I'm
very
sure
unnerving.
it never been for me. I'm like, where am I? I'm supposed to be
Where
talking.
am I? Yes, unnerving for you. All right. You're plugged back in though?
Yes. Can you see me?
Okay.
Oh, you're not sharing yet. I don't
We're
share
not
yet.
sharing yet, no.
Okay, I got you. Back to where I'm at. Well, I won't share the right thing I want to share. There we go. Uh, okay, here, leaving off there. Um,
Perfect.
Okay, getting to the meat of the thing that I really wanted to talk about now, finally. This is our study, and so it is a collaboration. Save Hope actually developed this technology, but we are their partners. This is a CIRM-funded grant. Here are some of the investigators that are working. We have our site PA, our Dr. Smith, of course. Michael Choi is from our Hemonc division, and he's one of our on our cellular medicine service and looks after our CAR T patients. And I'm helping as well, seeing the patients. Karen Dubé is also doing some quality of work around, for all the subjects or all the participants are going to be having some key informant interviews. Why this came about is we have the Stem Cell Institute, and so this gets to sort of what am I up to now? I now run this one of the centers within the institute. So this is based on a $150 million gift we had over 5 years to deliver new regenerative medicine approaches. So I mainly helped with the clinical trials arm of this, which is the Sanford Stem Cell Clinical Center. My assistant, my deputy director is Sandeep Patel, who you all might know as one of our oncologists. We also have 5 other centers. We have ones that are more basic science. We have ones to try and develop new innovative products that would be launched into maybe companies. We have a number of space-related things. We were at SpaceX yesterday, and we're trying to develop more space. We've had 14 missions in space with stem cells, organoids, cancer, et cetera. These are the studies we're supporting right now and at the clinical center. So you can see it's a wide variety of things. Gene therapy for cystinosis. We've done a liver study that was part of my study as well. We have some new studies for Parkinson's disease, stem cells for Parkinson's disease. We have for focal episode, which have been a very successful study so far. Spinal cord injury. We're working on ones for MS. And then we have a lot of cancer studies. So that's why a number of these are actually CAR T therapies as well that have been used. So, again, showing that experience that we have. So the product that we have that we're going to use in our study uses CMV cells. So and these again developed through City of Hope researchers, but kind of on the premise that there's a lot of immune cells that react against cytomegalovirus, and so we've heard a lot of cytomegalovirus. That's one of our favorite viruses here, what I've worked in the past, and Dr. Gianella. Dr. Smith. So, that it makes sense. There's a lot of these CMV reactive cells. They're there to, try and damp down cytomegalovirus, which is one of the herpes virus family of viruses that we almost all have, and that they will constantly, as we know, we can get the herpes outbreak. Well, same thing, CMV can come back when your immune system goes down. And of course, back when we didn't have enterococcal cytomegalovirus was a major problem for those people with very low CD4 counts. So what we do then is that we isolate those CMV sort of reactive T cells, and then they are manufactured to have a different BNAM target. This one in particular, N6, has a much higher, broader sort of neutralizing capacity than the VRC01. So that's why we're more hopeful that's in this particular study showing them 98% of neutralizing power with this particular antibody. So the idea would be that we'd have these CMV reactive cells, we'd add in the ability to fight off HIV. And there's a lot of potential with that. But could you stimulate that more with the CMV vaccine or other types of strategies? But right now, we just got to show that you can give these cells to people and that it's safe and that shows some, promising signs. And here's kind of the process of those cells. Again, which I had outlined earlier, we have new participants that would have leukapheresis, have isolating those CMV responsive T cells, and they're predominantly CD8 cells. And then they'll be reprogrammed here with the N6. And we also, and there's on the right-hand side, you see that the structure of the receptor there, we also have that co-stimulatory binding area, the CD3 zeta chain part, and then the new N6 BNAB. So this is sort of a similar to that study I showed you, but we're using the more broadly neutralizing and more, if you will. Once that, we're during their leukapheresis, they take a short break in HIV medications and their ART, and then they go back on their IRT, and then during the process of making the cells, they're also expanded, here under the influence, in the presence of darunavir and fluvatride, just to prevent any HIV infection of the new transduced cells. So hopefully we come out the other side with this new CAR T therapy that would be then be the first in human for this particular product. But it's been, gone through a lot of preclinical work. And before it took us a little time to get it up to speed. We got the funding. The cellular therapy lab is now up and running at City of Hope. And in fact, we've already enrolled our first few patients. But here's the sort of process for those patients. We are doing a consenting here at UCSD. So we are the core for the recruitment side of this. And then we're doing the screening. There's a number of things which I'll show you in terms of the inclusion, exclusion that people must meet. Generally, they have to be healthy people on stable antiretroviral therapy, fully suppressed with higher CD4 counts. Then they have this 4-day break, apheresis. If that fails, and I talked about, you saw that we only got that sometimes you can't make the product. So if it fails the first time, there can be one repeat apheresis done, and if that failed, then they wouldn't go forward. But there is a potential that if they're unable to make the product, that we can try it once more. This is I guess it's probably the next slide, but it's a standard 3+3 design safety type study. It starts with sort of a small dose, which is we don't necessarily consider that therapeutic dose yet. That's the 5 × 10⁶ cells. The EGFR part is just a marker so that we know if the cell is still there later on. So it's just a blind marker for the cell that's been added in the manufacturing process. And then we have what we consider to be 2 dose levels, 25 and 50 × 10⁶ cells. And then basically then it's all about monitoring for dose limiting toxicity, and then other markers to see if there's any sort of response there. And then they go into this long-term follow-up phase where we would continue to see them here over the next 15 years. Um, so yeah, so it's they only get single dose. It's 3+3 expansion escalation. So if you're not doing a phase one, most of you probably are. Basically, you do three patients, and in this case, and as said, requirement of FDA, and for good practice, would be that each person is done sequentially. So after that first patient, we're going to wait 60 days for safety assessment before the next patient would go through the procedure. So even though we maybe enrolled two, maybe even a third person was coming, they can't go in. We're going to have slots over the next year. I think the first one starting in the fall, and then they'd have to get through their 60-day assessment. And then, if safe, then we can go forward with the next patient. With that, if we reach the whatever the final, so usually with a 3+3 design, what happens is if the one patient has a dose limiting toxicity, then you'll do an additional three patients in that group. And then if all three are fine, you still move up to the next dose. If you have another event, then that is the end, and that would be considered the dose below it is the dose that you would use. But usually the idea would to do six participants at that maximum tolerated dose, and then there's potential to do another six participants after that, so up to a total of, well, 15 or so people. This is those inclusion criterion. They have to be adults with a good Karnofsky score, maybe suppressed, and CD4 is over 450. We're going to do, just the basic labs. They have to do a echocardiogram. They have to have want to participate in this. Obviously, can't allow any sort of pregnancy, breastfeeding in this, and have no other active, infections that we'd be concerned about. Because again, there's a point here where they could be immunosuppressed. We don't want to have new infections come up. Our primary outcome is really safety. And that so there's dose limiting toxicity are really more grade 3 and 4s. And if you have persistent grade 3s, or if you have grade 4s or 5s, then it stops. They'll otherwise capture all other adverse events as well. But then some secondary are really important because, of course, we want to see, is there any benefit that we see with this? So we want to see if there's increase in CD4 count, if there's any changes in just measured virus. We want to measure that those cells persist over time and whether they go. Do we have any cytokine, types? What changes in the cytokine profile? And whether we get some memory of these T cells over time. Because again, the long-term goal is that they would persist and be able to continue to be reactive. So we also have a side study. There's a qualitative study with Dr. Dubé, which we'll do some 2 in-depth interviews with them about following screening, and then after, getting their treatment. Some, main high-level concerns about the study. Obviously, this is a first-in-human product, not unlike doing any other first-in-human product, which is something we do a lot actually in my clinical center. Most of our studies are first-in-human studies for a lot of our stem cell and regenerative medicine products. So this is something we're quite used to doing. We have to go, as I said, very sequential and very methodical approach in terms of for safety and to moving sure the first patient is safe, the next patient, and the next patient. It's really this, and I kind of alluded to the cytokine release syndrome. And the worst case scenario is that people have passed from that in the past, but never with any of the HIV related studies. And that's why it complicates it because we have to watch people very closely. And this requires people to stay at City of Hope for 14 days. So product is being made there. Once we have screened a patient, they would go to City of Hope to get the apheresis, come back, and then go up to get the actual infusion. And then they would stay in their local, they have a facility there. The City of Hope does a lot of bone marrow transplants. So so they're used to having people stay there with them at a like a hotel that's there. And then they would be seen daily to be closely observed during the riskiest time of post CAR T therapy, which is those first 14 days. And then they would come back to be observed with us. And of course, we have those facilities. And I thought I had a slide there, but we have We see all our patients in the Komen Outpatient Pavilion up on La Jolla campus, adjacent to Jacobs Medical Center. And we also have designated beds that are cellular medicine service beds. And those are where all our CAR T therapy people are seen. That's what Dr. Choi is there. And we have actually a whole team of people, of nurse practitioners, physician assistants, and other physicians that run that service. There's also a new thing with CAR T, and we're not sure if this kind of CAR T has this, but actually, there's somebody we actually knew from way back who's in, I think, Australia, but he found just in this past year that there's some CAR T related lymphomas that are pretty hard to treat too. In the currents, when you bundle together all the studies, there's about a 1 in 1,000 chance of this lymphoma related to the actual CAR T cell itself. Because this is where that, getting too eloquent can be a problem. Because every time you do CRISPR, when you do new gene editing, you can also hit off target pieces. And so the concern there would be, are you creating gene changes that could be precancerous? Although I'm not sure that was the mechanism in this case, but that certainly is the concern. That's why the 15-year follow-up as well. Oh, here we are. This is what I was talking about, is sort of where everything gets done. Here is on the upper pictures, you see City of Hope Hotel. And then down below where we've seen here at the Komen Outpatient Pavilion and the Jacobs Medical Center, which you've probably have all driven by or seen or been in at some point.
Sheldon, just letting you know that it's 8:47. There are a few questions in the chat, so I don't know how much you have left. Just FYI.
2 slides. So this is just the participant, what that looks like for them. They also need to have a caregiver go with them with City of Hope so they can keep an eye on. And so they've been also planned that they would all be allowed to stay there and be reimbursed as well through that time. Still some questions. This is my last slide. If this is going to work, what is it need? I mean, I think that we're going to need to have, can they persist long term? I've talked about that a bit. Can we hit all the reservoir? That's another big question. Can it actually find all those? Can the CAR T avoid cell HIV infection themselves if they are T cells? That's another question. And then how long do we have to be undetectable before we say they're cured? Would be still, we haven't got that far with any of these CAR T, but obviously with the other ones. And that's it. We're going to questions. Marvin is our contact for our recruitment activities, if anybody's interested in that. So what questions do we have, Jill?
I'm going to start at the top. So for Michael Luella, what are your thoughts about the Geneva patient who has been included in the roster of people being cured without the CCR5 Delta 32 mutation?
Now, did they get a regular stem cell transplant? Is that what they were? I'm not.
Yes.
Yeah, I think what there's a few points where you can, as I see it, if people are early in their disease, particularly, they may not have a bigger reservoir as other people. So if you got rid of the reservoir with just the treatment of the chemotherapy and then you had transplant, that is potentially cured. And I think that's why they did that's why it was even interesting to see that approach. I think the longer a person has had active HIVs, there is more chance that the virus can have reservoirs in different places anatomically that may be preserved following ablative therapy. So my take on that would be that it worked because we got rid of all the reservoir with the ablative therapy. And then it didn't matter that you needed a CCR5 release because you didn't have any virus hanging around. But all it needs is a few cells that would be, that where the virus can come out of latency and reinfect those cells. So, and I think, I don't know how many you've seen this, but I've seen people that were sort of long-term suppressors, off ART, no viral load, and they are 10+ years out, and then we see HIV come out, and they can't believe it, right? They can't believe it. They go, "I was cured," and so there's a long time. And so have we just not watched them long enough? And if they are only heterozygous, they're going to have still some CCR5 showing. And that'd be the other thing is, are they expressing that CCR5 enough? And I imagine even without CCR5 being deleted, it would be theoretically possible that you could have a cure if you could get rid of the reservoir. That's kind of bottom line. Would they be immune to
There's,
reinfection? No.
yeah, there's some, right. Sorry. Glad you're seeing that.
Because people that are no heterozygous, as I understand it, with delta CCR5 or delta 32 heterozygosity, will still have CCR5 and still can get infected, but their chance of infection, their probability of infection is lower. So they have some resistance to new infection, but I guess also depending on how much, I would think if that person also had ablative therapy, they don't have a lot of memory for HIV, but it'd be if they did have any leftover immune cell, they would have some memory to some HIV, but still they could still get infected. Bottom line on that, I think.
Okay. Um, Dave Gerlotti, let me make sure I didn't skip anyone. Dave Gerlotti asks about the long-term follow-up. Participants move, funding ends, researchers retire, science advances. How are all of these considerations reconciled with a mandate for 15-year follow-up?
Yeah, it's tricky, right? So and this is I think something that has not been fully fleshed out. Now we have a plan. As the Stanford Stem Cell Clinical Center director, what I've done is I've initiated a registry for everybody coming through our clinical trials that we'll continue to follow them every year. Right now, that's a phone call because it's not an unfunded thing, but we're trying to build that into a longer term follow up for everybody to look particularly at, say, maybe malignancies and stuff like that. But if the company is, because we've had actually from previous ones where a company went bankrupt and they had done a stem cell treatment and they don't even exist anymore. So that's a problem. And then, as you point out, usually you have a 5-year maybe funding cycle. What happens after that? So and now for our gene therapy study, this is the same issue. Funding ended. We did help with that study. Those patients need follow-up. And so they were able to secure funding. The product itself was pretty successful for cystinosis. So it was bought by a pharmaceutical company. And eventually they were able to get financing for investigator-initiated study to continue that follow-up. But this piecemeal way is not particularly good. But I think at least here, we have a plan to do it because we are going to continue to do it as long as we can. But and then will there be a long-term solution? You just can't fully ensure that if unless there's some sort of way that say a company has to put in so put aside money that would pay for the full 15-year follow-up. What else we got?
We got our side effects related to effectiveness of the therapy.
Yeah. To some degree, yes. I mean, so when we think of cytokine release syndrome, it's really the mass effect of killing all these cancer cells at once, and that stimulates this big cytokine response. So, yeah. So what if you had a massive reservoir of HIV and you had massive cell release? Yeah, that could potentially be, I would think, more likely to have adverse events, but I think the scale of it is much smaller. So, who's going to benefit? I think getting back to that Geneva patient, people that are sort of like earlier in, I don't know where they were in infection, but that the concept that if they were earlier infection picked up in an acute infection, like we've done so well with in San Diego, that probably limits the reservoir and where how many places we have to get to. I think they'd better be better cure candidates.
Susan Little asks, I recognize that this is entirely a safety study, but ultimately, what are the measures of antiviral success that would be used? Yeah, other than persistence of CAR T.
Yeah, so I would say that with other studies, the way you do it is you look at what is the response? Are you seeing reductions in proviral DNA, HIV? Are you seeing responses of improved CD4? And then you could, in my mind, think that, for some of those patients, you could do a follow-on study where they would have a planned treatment interruption. And so I think that would be, you'd have to set criteria based on the laboratory data that would then lead you to attempt the treatment interruption, and see what, as I've shown with some of the other slides, would there be a viral response.
And sorry, I didn't see, please feel free to raise your hand and unmute yourself. Jeff Taylor had written or wrote down an idea about handling the 15-year follow-up, maybe to create a standalone registry into which people would enroll.
And that's what I'm doing. I've already got that actually approved to do a standalone registry for all every patient that comes in, not just to this study, but every stem cell study or regenerative medicine study, so that because I could foresee that I want to know as the investigator broadly, what is are these things working? Do we have long-term survival? Do we have long-term side effects from these? And I'm not just talking about HIV cure. I'm talking about, our stem cell therapies and gene therapies for these other diseases. How do those patients do? So, but and I think down the line, maybe we can discuss with a lot of our funding comes from the California Institute of Regenerative Medicine. So, is there some more direct funding that could go towards having a more robust follow-up program that would be, as you point out, like a standalone registry for everyone across. We have actually, I think, nine funded, we call alpha clinics. So the alpha clinic is actually the CIRM funded unit that is the kind of the clinical trial unit that sits within my clinical center, because there's other components to that. For example, I have something we call the START program, which is like we have four program people that know really how to do FDA applications, and they're really helping the investigators do the preclinical work, apply for INDs and stuff like that. But then the alpha clinic part, which is really the clinical trial unit, is funded through these, and there's nine of those. And so collectively, we want to do this. We're also trying to work collectively to be more community engagement. So we've started a whole community engagement program in my center, and trying to build collaborations with other regions. And then CIRM is trying to set off an independent unit that would actually assist participation by, travel, other types of things, two types of studies, and that sort of something's going. So yeah, there's sort of these layers of things that we're trying to develop. So this makes it more accessible for everybody, and that would have a higher level of, doing what's right in terms of follow-up.
Sheldon, that was actually going to be my question. I know we have established community advisory boards within the CFAR and the AVRC. Have you engaged much with them, or it sounds like maybe you're setting up your own community advisory boards. What's that looked like?
Yeah, I think that we would be. I think we have had a community advisory board that I didn't attend, but we're going to follow back on that. We also have a community advisory board that we formed for the clinical center. So there's going to be some, a follow-up. We were actually kind of and yeah, Jeff makes the point that he's been on the representative for the protocol, and that was built into the actual funding for it. And Jeff, I don't know if
I
you
just
want,
wanted to make sure. Thank you. No, that's
maybe we
great.
can ask Jeff to speak to that. I mean, what's been that perception and of how how this has progressed?
And Jeff, you have 1 minute since it's 9 o'clock.
Okay, I'll talk really fast.
Sorry.
Well, and briefly, I mean the AVRC advisory board has been the one that's been advising the study. Basically, it went to the ACDG briefly, as you know, then got cut. They did have a representative there, but so it's been the AVRC, which I think does a good job. They know the issues and understand it. But as we go forward, it might be interesting to have a stem cell advisory board as well across different disease areas.
Thanks, Jeff. And thanks, Sheldon. This was great. Only one hiccup along the way, and I think you rebounded very quickly. with a computer shutting down on you. This is really exciting that this study is happening, with UCSD and City of Hope, and I think there's a lot of excitement around this. So we'll definitely have to have you back when we have some data or some patients enrolled, and we appreciate you coming and sharing with us.
Thank you. Great to see you all.
Have a great weekend, everyone. We'll see you next week.